Right, so if you're drinking a glass of milk, you're not keeping that milk in your mouth very long. You're not going to be taking lactose and breaking it down. You don't even have the final step enzyme, but you're not going to be taking any polysaccharides and breaking it down to lactose either. All right, most of us don't chew milk or swish it around in our mouth very long. Even with crackers, remember I said as you chew long enough, they would start to taste sweet, but most of us don't do it even that long. So it's a limited amount. So it does occur, but it's time dependent for the amount of time you have the food in your mouth. Although we do have salivary lipase, there is negligible amount of fat digestion that occurs there. And we have no enzymes that even pretend to work on intermediate digestion of proteins. And even with the carbohydrates, again, none of that's going to be absorbed because we don't have any final steps breakdown. Okay, in the esophagus, for the few seconds it takes you to swallow, there's a continuation of whatever got started in the mouth. But we don't add any additional enzymes. The only glands that we have are in the distal region of the esophagus, and those are mucus glands. They don't contain any type of digestive enzymes. And then we enter the mouth, the stomach.
So when you say activity, that's, you still got amylase, celery, and...
Yeah, for the few seconds. Just what was in the mouth. So if you chew the food and it moves through the esophagus, it's still going to have those enzymes mixed up with it. So for the few seconds it takes to enter the stomach, that's going to occur. All right, now we get to the stomach. So here's a view of the gastric pits. These would be the surfaces of the goblet cells, of the columnar cells. And remember the gastric pits line formed in the mucosa. And you can see some strands of mucus produced by those mucus cells. And pH of 1 to 2. Why is that? Why do we have such a low pH? What cells make hydrochloric acid?
Chief cell?
No, the parietal cells. Those are the ones that have the pink color. All right. So parietal cells are secreting hydrochloric acid, and hydrochloric acid functions to activate pepsinogen, which is made by the chief cells, to the active form pepsin. Okay, so these are the chief cells making the pepsin, and the pepsin is an intermediate enzyme for protein digestion. So I'll put some. It's a little bit more than negligible, but it's not the major site. I was looking for some videos to show digestion, and I just saw your email, but I had during class time, so I haven't looked at it. And I thought I found a really interesting one, but it said all the protein digestion occurs in the stomach. I'm like, that's wrong. And there were a couple of other errors. So I thought I don't want to go through correcting everything and then have students walk away. They didn't hear the correction, they just heard the video. So I decided not to show it. But we do have an intermediate stage digestion enzyme, and it works only at an acidic pH, all right? So there will be some, but it's not the major site. It's not the characteristic location where we break down protein. With the acidic pH, are we continuing our salivary amylase? No. So for carbohydrates, the stomach does not produce any enzyme that works with carbohydrate digestion. So, and we stop the salivary amylase. We denature it because of the low pH. So the salivary amylase is no longer working. The stomach itself does not provide any enzymes, and so none. stomach does make, also by the chief cells, the stomach does make gastric lipase. But again, that's negligible. It's similar to the mouth. So fat digestion in the stomach is negligible. The fat isn't such large, it hasn't really been emulsified much, and so our surface area and timing is just not sufficient to do much digestion. Okay? So very little, but again, even if there is some protein digestion, it's not absorbed because it's only intermediate stage. Okay? All right, so what activates digestion in the stomach? When you start to feel your stomach growl, when it starts to rumble and stuff, what's going on? That happens before food even actually enters into the stomach.
Isn't that something Vegas?
Exactly. So the next chart here looks a little complicated just because there's so many words.
Quiz last night would have been much more beneficial to this class.
With this one, on the, well, there should have been a physiology part to
There
it.
was no picture on some of them.
There was
Me
no pictures
too.
on it,
Oh,
but
okay, somebody, that one I'll go like the male reproductive, go back in and look at it. So on the excitatory aspect to it,
that helped me answer.
you will have, we're going to focus on these two components right here. All right, so smelling food, thinking about food, you're stuck on an island, don't have anything to eat, or you have to take a blood test and they told you to fast for 12 hours before that, and when you wake up, that's all you can think of. So that cerebral stimuli, or just having the presence of food on in your tongue, but not in your stomach, is the cephalic phase. Okay? And the vagus nerve is our nerve for the stomach, and so it will activate, it will release acetylcholine, and through a couple of different, I have another series diagram here. So the vagus nerve will activate the submucosal plexus of nerves, used to be called the hour box, if you've heard that term before, and will cause the parietal cells and the chief cells to secrete the pepsinogen and hydrochloric acid.
Got that one wrong.
Okay? Now, in addition to that, remember we had hormone cells I wrote about on the board? The enteroendocrine cells. And I think, Libby, you were the one that asked if they were the same as G cells. Somebody did. And they are, because the G stands for gastric. So we have both nerve innervation causing the increased secretion. Gastrin is a hormone. So this is done by the G cells. It makes gastrin. So here we have pepsin, which ends in IN, but that's an enzyme. Gastrin ends in IN, but that's a hormone, okay? And gastrin gets in the blood, goes throughout the body, but has receptors back at the stomach. So it has to come back to the stomach to find receptors, and then this hormone and its receptors are going to go back and increase activity of the chief cells. They also bind to the smooth muscle of the muscularis externa and increase the activity and increase the churning effect. So that's the phallic phase. Now you swallow the food. You chew it and you swallow it, and it gets into the stomach, and we start to distend and stretch. The cells actually have contact with the food, and so in addition to the vagus nerve, we add the myenteric plexus. My refers to muscle. So this is between the layers of muscle, the inner circular and outer longitudinal, and we just enhance everything. So it's just another snowballing, if you will. And so this is being secreted, the muscles are contracting, churning is going on, and we mechanically break down the food, do a little bit of chemical breakdown with protein, and create a substance known as acid chyme. Okay, so chyme is that turned up food, partially broken down, that we have in the stomach.
Or if you drink too much, what you end up expelling.
Yeah. So now that moves into the small intestine, okay? And before we look at the products and what's produced, let's reverse the sequence here and look at the control. What happens? So on Friday, when we talked about the duodenum, remember I said that it was a receptacle. It was too short for much digestion to occur, but it receives the chyme from the stomach. It receives the bile from the liver and gallbladder, and it receives the pancreatic juices, which are bicarbonate ions, and digestive enzymes. Okay, so that's kind of being shown here. I'm going to re-diagram it on the board, and this is your extra credit for the digestive system. It's this sequencing of excitatory and inhibitory influences. So we start with our stomach.
Professor,
Yeah.
does the parietal cell secrete hydrochloric acid only when there is food, or does it?
Well, with smell, yes. Okay, and also that's the stimulus for the release of the hydrochloric acid. I'm not saying they don't do it other times, but that's when there's an increased amount.
So is it like with the more there is food, the more hydrochloric acid is going to be produced?
Especially if it's protein. The protein type of food is compared to a lipid, a fat, or something of that nature. Before we leave that, I mentioned it in the morning class. Those of you that have had microbiology, what is the bacteria that's now associated with stomach ulcers? Helicobacter. Pylori. All right. This is an interesting illustration to me of how we think that we're so modernized and open and exploring new information, and we look down our noses at people in the 1700s and 1800s that didn't know what caused disease, and the idea that a doctor would have to wash their hands as they moved from one patient to the next was, you didn't want to wash your hands, that would make you sick, kind of a thing. And it started a lot with women that had given birth. They would be carrying infection from one mother to the next. And somebody started to notice that and started encouraging washing hands, and was kind of laughed at by the rest of his profession. Similar process here. Back in the early 80s, we had a doctor in Australia notice that, just by chance, serendipity if you will, that patients of his who had ulcers, that he was providing antibiotics for other health conditions, their ulcers improved. And being a curious kind of a guy, he noticed that, then started to experiment with other patients that had ulcers that did not need antibiotics, and started giving them antibiotics and noticed their improvement, and started investigating why. And he would go around to various medical conventions and started giving talks about his theory that bacteria were causing, their excretions were the cause of ulcers. Because when he got rid of the bacteria, the ulcers also healed. And he was laughed at. We're talking in most of your lifetimes, okay? And if not, then I'm feeling really old. But, so we had he hadn't quite yet found the bacteria, but people were saying, bacteria can't live in the stomach, it's too acidic. Well, we know now that the mucus actually protects them, and they colonate underneath the mucus. It got to the point where he was not just not believed, but when he would submit his paper and his talk to a medical convention, he was denied access. They're like, we don't want to hear that anymore. So he would lie. He would say, well, I'm going to talk about the effects of antibiotics on foot ulcers. And then he'd get up and start talking and revert back to his stomach ulcer theory. Eventually, a few more doctors started listening to him. A few doctors from the United States traveled to Australia and worked with him for a while. And gradually, they were able to prove his theory was correct, and now it's the accepted cause of most ulcers instead of type A personality or stress-induced, which has an effect on it, but it's not the original cause. So I just wanted to mention that before we look at this control. And recognize that we're not as modern and open in thought as we try to think of ourselves. All right, so from the stomach we have acidic chyme. And that's going to flow into the duodenum. From the liver, and stored in the bile. We have bile salts. So remember, the gallbladder doesn't make it, but it stores it, whereas the liver actually synthesizes it. And then in our pancreas, we have our pancreatic, our digestive enzymes, and bicarbonate ions. Okay, kind of lay out the background. So as food enters our stomach, that is going to stimulate. When your cream is this color, you'll know digestion started to take place. It's going to, so food thoughts of food. I'm just going to put cephalic phase here. Both of these are going to be excitatory to muscle activity, motor and secretory activity. So we're going to see increased motor movement, we're going to see increased amounts of enzymes, hydrochloric acid and pepsin, we're going to see increased amounts of gastric. Okay, that's what we covered in the cephalic and gastric phase. Now as that chyme moves into the duodenum, especially if it is has proteins in it. So if our chyme is rich in proteins, actually lipids, proteins and lipids, but here we're looking at lipids primarily. Over here. That's going to stimulate the small intestine to produce a hormone known as cholecystokinin, CCK. That's a hormone. And cholecystokinin is going to go to all three organs that I have diagrammed up on the board. It's going to get in the blood since it's a hormone, and it has receptors in the stomach. So if we take cholecystokinin and send it in the blood to the stomach, is it going to be excitatory or inhibitory? It's going to be inhibitory because we don't want the pyloric sphincter to dilate again and release more chyme until we've taken care of the chyme that's already in the small intestine, in the duodenum. About 1 to 2 tablespoons is released through the pyloric sphincter and into the duodenum, and we need to neutralize it. We need to add bile salts to it. We need to add bicarbonate ions and digestive enzymes. So cholecystokinin has an inhibitory effect on the stomach for a few moments, for about 15 minutes or so. and slows it down. You can tell by its name that cholecystokinin, a cholecystectomy is the cutting and removal of the gallbladder. So cholecystokinin also has receptors in the gallbladder wall that causes it to contract. So it is excitatory to the gallbladder. And that causes the bile, with its bile salts, to move down the common bile duct and be added to the chyme that's in the small intestine. All right, in addition, cholecystokinin goes to the pancreas and has an excitatory, stimulatory effect on the release of digestive enzymes. So now these enzymes are moved at a higher rate down the pancreatic duct and into the duodenum. All right, the presence, yes.
So the digestive enzymes, is this insulin or?
No, that's hormones. The digestive enzymes are pancreatic lipase, pancreatic amylase, trypsin and chymotrypsin, what we're going to talk about in just a moment. All right? So the lipids cause the small intestine to release cholecystokinin. The presence of acid, which is present anyway because it comes from the hydrochloric acid of our stomach, is going to cause the small intestine to release another hormone known as secretin. Let me put that in a different color. Okay. Secretin also is a hormone, so it's going to go into the blood and it's going to go to all three organs. So when secretin goes to the stomach, it does the same thing as cholecystokinin. It inhibits the stomach and slows it down. When secretin goes, it does not bind to the gallbladder, but actually affects the liver and causes the liver to actually synthesize more bile. Both of them have the same effect. There is more bile entering into the digestive tract, but in the case of cholecystokinin, it's going to work more rapidly because the bile is already made. Okay.
One more time, Barry.